introduce her.
Absolutely. Can you hear me, Katie? Okay. Hi, everyone. I'm Divya S, and this is my second summer being a Magic Mentor. Since I did my undergrad degree in biology, I'm always looking to work with students on science-related projects. And so I was really excited when I got paired with Daisy Flatman this summer. She worked on carrying out a chemical reaction with potential real-world applications that I'll let her share more about. But at our first meeting, I was already impressed because she was fully prepared with a really thorough document listing out not just ideas, but also materials and logistics for each one of those. On top of that, she has also been so proactive in driving her project forward and asking thoughtful questions and guidance when needed. Her experiment involved a lot of coordination and buying some pretty niche materials and supplies. And so while that resulted in its fair share of challenges, Daisy was unwavering in her determination to find a way to make things work. And she really ran the extra mile to ensure that she could find a way to complete her project successfully. We also had a lot of fun along the way, and I particularly enjoyed the special appearances that her cat Cheddar would make during our sessions. Daisy is driven, intelligent, curious, and hardworking. I'm excited to follow her future trajectory and see all of the great things that she's going to accomplish. But for now, it's been a real pleasure being her mentor this summer. So I'm going to pass it on to her now to share more about her project.
Okay, should we jump? Oh, so I'm in ninth grade currently at the Ethical Culture Fieldson School in the Bronx. And if we go over to the next slide, I can talk a little bit about Divya. So Divya got her bachelor's degree in biology from USC and then her master's in business analytics. So currently she's working for Intel, but she still advocates for having interdisciplinary learning between biology and other kinds of technology because they all kind of intertwine. And she loves to paint and read and travel to some of the places she reads about. And while she currently lives in the Bay Area, she grew up in Southern California. So if we go over to the next slide, I can talk a little bit about the project. So isothermia is basically because the main technology behind my project was loop-mediated isothermic amplification. And my goal was really to create a kit that somebody could use at home to test for a specific gene, especially because currently there's a lack of commercial kits on the market, while some are like really expensive, between $80 to $150, and they don't produce results very fast. It could take like between 3 weeks to a month. And another big thing was that you have to send your test to a lab where then they can make results. But I was interested in seeing if how a person could do it at home. And this was really helpful for me because I was already interested in like genetic testing and traits. And I was able to kind of take that interest and put it into something that I thought could have a bigger impact on the world like around me. So if we go over to the next slide, I can talk a little bit more about the details around the project. So as I said, one of the main technologies I used was LAMP, which is a form of genetic amplification that basically takes one gene and it will bond several primers to it that will create copies of that. And when it does that, it will form more phosphate bonds that will change the pH of the solution. So the main way that I saw to detect that was using pH changing dyes that would show that it would turn yellow if it was positive for the gene, or it can stay red if it was negative. I also had to end up designing a lot primers. And so we took the like genetic sequence and we just kind of like tinkered with that until we got one that wouldn't like bind to itself and like was the fastest, I guess, is working. And even then, I still would like to like look at more of those and see how I can make something better or make something for potentially a different gene. And so some of the main things I did were compiling like a list of materials I needed, which, as Divya said, were kind of hard to get because not a lot of companies want to sell enzymes to a 15-year-old. I also had to find participants, which I guess in this current like age, people are more protective over their own DNA and stuff like that. And I kind of had to see how everything would work together because there's just so many variables, like keeping like the area sterile and stuff like that. And in the future, I'd really hope to expand to like other genes and other areas, and also making it more consumer friendly, because really what I did was trying to execute it at home myself. But if I wanted it to be more like accessible to other people, I'd want to like find ways that I can make it easier, or like have like a more sterile environment at home without necessarily needing a lot of materials. So if we go to the next slide, I can talk about my magic experience. So the highlights were really having a person to like ask questions to or guide me through the process, especially because I don't really have a lot of experience in chemistry or biology. So it was really helpful to have someone who could answer those questions and had a background in those sort of things. Um, and the main challenge was really getting my materials. That took like probably like 4 weeks on its own, because I just like kept talking to companies and trying to find different things. And I sent a lot of emails to customer service representatives. Um, some of the main things I learned were like lab techniques and stuff like that. As I said, trying to keep the area sterile, even though I'm at home. And my cat has like a really excited to jump on tables and stuff like that. So I had to try to keep my dog and cat away from that. And I really just tried to like simplify the process, because a lot of the information is kind of made for like a lab area, and I don't really have access to most of those things. So I just tried to like break it down into the simplest way that I could do it. So if we go to the next slide, I can show you my demo. So these are just some images of some of the components in my project. You can see the first image is just the different samples, and this was like my first attempt, and you can see that like the color change between the second one, which is my mom's, who I knew already like had the gene, so she basically was not a control but a way for me to see if it like actually amplified the sequence. And the color on these isn't really what I expected it to be, because I think I got the pH a little bit wrong when I was designing my buffer. And it's more of like a purpley color, so it was more basic than I wanted it to. And then the middle, you can see just the different variations that it could be. So on the far left, that's red. That's kind of what I want it to be, because that's kind of like the neutral color. And then on the right of that, you see the kind of purpley color, which is a little too basic. And on the right, that's yellow and it's too acidic. And then on the very right, you can see my cat, who is my control, because the gene is only specific to humans. So I just figured that that was kind of the closest way I could get to it at home without having to like make fake genetic like sequences that didn't have it, which I would be interested in exploring if I wanted to continue the project, because I don't think I can really put my cat's DNA in like 100 test kits. So the next slide is just, if you jump over there, it's just a little video of some of the techniques I was using. So the main thing was the colorimetric dye that I was using. And on the right, you can see that's pickle juice, which is more acidic. And on the left, that's bleach. So you can see how that sort of changes. And then one of the main ways I kept everything sterile was using this syringe filter, which was meant to sort of like not let any bacteria through. And then another thing that I had to do was heating. So I'd wrap the samples in tinfoil, and then I'd put that into my thermos, which then I would, so it wouldn't rise to the surface when I filled that with water. And I'd make sure it was at the right temperature, which was between 60 and 65 degrees. I'd fill that with water, and then I'd seal that up and then put that back into the water. So it would try to maintain like a good temperature throughout the whole thing. And I'd leave that for about 30 minutes. And then for the lysis solution, that was 15 minutes, which I experimented with different kinds of lysis solutions, which is what extracts the DNA from the cell. So the first one I used was lye, and the other one I used was just a mixture of some different things like tween and stuff that would take it out of the cell, but that required heating. So I was initially like interested in the lye, but then I had to switch over because I was getting the wrong pH and stuff like that. So I tried that too. And if we just jump to the final slide, that's just a little instruction manual that I made for if someone's to use this kit. This is kind of like an idealized version, which I would be able to like freeze dry things. So I wouldn't have to keep it in a freezer if I wanted to like sell it commercially or something like that. And it just lists like all the hazards, like what's in it, like troubleshooting if you like something goes wrong. and just like how the actual technique works, because I know probably not a lot of people know about like the specific kind of like amplification, and then just all the materials and just how you would follow through the process. I tried to simplify it the best I could, but I know there's probably some ways that I could make it better, I think, in the future. And that's all I have for today.
Wonderful job, Daisy. Thank you so much for sharing your project with us. What questions do we have from the audience?
That's
I have a
amazing,
quick question.
Daisy and Divya. Oh, sorry.
Go ahead.
Okay. Really great work, great idea and execution, and the presentation was really good too. My question is, did you research if there are some existing kits like this in the market?
Yeah, I did look at what other kits were available, and the ones that I saw were mostly for if somebody wanted to use the technique for like research in a lab or something like that. And what I was focusing on is if a normal person wanted to use this for testing for like a gene. And I guess that was the key difference between what was already made and what was I had. And I sort of looked at what was in their materials and tried to model mine off that, both like seeing like what they did and seeing how I could change that for someone who was going to buy the kit and make it easier to use.
Perfect. Really good.
I just wanted to ask real quick, I don't know if I missed what gene you were testing for?
Oh, I was testing for the ACTN3 gene, which basically like allows you to produce muscle mass faster and like breathe in air quicker than someone else. And I thought that would be like the best gene to test, because while like in the future, I'd be interested to seeing like what would be predisposition to like different cancers and stuff like that, because I think that's like one of the most useful ones in like what I was doing, which was I was trying to have like testing my friends and stuff. I just thought it would be easier to have something that like didn't have a huge effect if my test worked or not. And because I wasn't sure that I'd have the most reliable results and stuff like that.
Super, super cool.
Yeah, I just have to say, my mom's a geneticist and has always talked about this being the future rather than the multi-$100 kits. How did you stumble upon the lamp technique over PCR? Like, it's super cool that you found something you could do at home.
Well, I knew that with PCR you had to like cycle through a bunch of different temperatures. So I just kind of like researched into like all the techniques that people were using. And I know that for genetic testing, they also use a mixture of LAMP and PCR. So I thought that like LAMP would be the easiest one to do at home, just because it doesn't require like that much technology that someone wouldn't have already.
Is it gene specific when they use LAMP versus PCR, or is it more like different companies using different things?
I think they use it for different genes depending on like what they're testing for.
Cool. Okay, thank you.
All right, wonderful.